Well, hello everyone. As I told you before, there's going to be now videos accompanied with the essay sessions that we're going to hold on Zoom. And I told you that this first video was going to be about chapter 15. As you may have seen, chapter 15 surrounds all the special senses, which include vision, taste, smell, hearing, and balance. So beginning with vision, we can see here the basic structures of your eye. What I want to make emphasis on is going to be the iris, which is the color in your eyes, it's a muscle. The pupil, which is just an opening. The lens, the ciliary body and zonule, which pull on the lens. And your retina. So I like to think of the eye as if it was a camera. And with a professional camera, you are able to adjust the amount of light that flows into your camera. And the same thing is going to happen in your eyes. The iris is the one that's responsible for this. As you can see here on the left, the iris is decreasing the diameter of the pupil, letting less light in. And this is going to happen every time you walk into a place where there's a lot of light. And this is what we call light adaptation. On the right side, you have the opposite scenario, where the iris is increasing the diameter of the pupil, letting more light in. And this will happen when you walk into darker places, and it's what we call dark adaptation. Also, with a professional camera, the entire plane of the picture is not going to be focused at the same time. It is always going to be focused on a specific object or person, and that is exactly the same thing that is going to happen in our eyes. So, if you wanted to look at something that is far away, as you can see here in the picture, the ciliary muscles are going to relax through sympathetic input, which causes the ciliary zonule to tighten and flatten the lens. Here we have the opposite scenario, where we would want to look at something that is closer to our eyes. The ciliary muscles are going to contract through parasympathetic activation, which causes the ciliary zonule to loosen up, allowing the lens to become bulgier and rounder. So what you want to think about is that far away means that the lens is flat, and close means that the lens is round. Now that we've discussed how we focus images and how light gets filtered in and out, we can discuss how we translate those images into an electrical current. So in this image, you can see a blown up section of your retina. And the most important thing here are going to be your rods and cones at the back of the retina. These are your photoreceptors. They are modified neurons that can absorb light and then translate it into an electrical current. So, as I mentioned, we have two types of photoreceptors, rods and cones. In this image, the rods are represented by the yellow cells and the cones are represented by the blue cells. Rods are non-color vision, which is basically your night vision. So when you are in a dark or a dim lit space, you can see different shades of gray that help you make up the world around you. And because it is a dark space, they need to be highly sensitive so that they can take in all the little amount of light that it's available. They are very numerous, there's about 20 rods for every cone, and they are mostly found in the peripheral sides of our retina. On the other hand, we have cones. Cones make up your color vision, and they have low sensitivity because they are active in places where there's a lot of light. They are less numerous, and they are mostly found in the central retina, which is basically all the space that is close to the optic nerve. Now, within our rods and cones, we have something that is called visual pigments. These pigments are formed by proteins called opsins that are bound to a light-absorbing molecule called retinol. We can find one pigment in the rods, but there are 3 different pigments that we can find within the cones. And each cone is going to have a different type of pigment. It's not going to have all 3 of them at once. And each of these pigments is going to absorb a different wavelength of light. And this wavelength has to be within the visible spectrum. So blue cones are going to absorb wavelength that is around 420 nanometers. Green cones are going to absorb wavelengths that are around 530 nanometers, and red cones are going to absorb wavelengths that are around 560 nanometers. These cones can absorb only this type of pigment. So when we see intermediate hues like pinks, let's say turquoise or aqua, we see a combination of two different cones firing at the same time at different intensities. And this is the way that we see different colors, and we don't just see red, blue, and green. After the photoreceptors have absorbed the light, they are going to transform this stimulus into an electrical one that can travel into the brain for it to be processed. This is a little bit more of a complicated situation, so I will probably discuss this in the next SI session with you guys to make sure that you're actually understanding what's going on. Now we can move into taste. The taste buds are going to be the receptors for this sense, and they are found within the papillae. This picture signals 2/3 papillae. So the first one's going to be the fungiform papillae. They are found all over our tongue, and they have around 1 to 5 taste buds, which is not a lot. Then the valid papillae, we have around 8 to 12 at the back of our tongue, so they are not as numerous as the fungiform papillae, but they hold so many more taste buds compared to the fungiform. And then we can find the foliate papillae at the sides of our tongue, so it's those little ridges that you can see. And they are very numerous in children, but as you grow up, you kind of lose some of them. Now, on the right of the picture, you can see a blown up version of a taste bud. Each taste bud is composed of 50 to 100 epithelial cells, which can either be gustatory or basal. Gustatory epithelial cells are our taste cells. These are going to serve as our receptors. And they have a long microvilli at the end that is called the gustatory hair. This part of the cell is very sensitive to chemicals. And the reason I say chemicals is because taste is a chemical sense, and it can detect chemicals that are dissolved in our saliva. So basically, how taste works is when you eat something, the chemicals in the food are going to be dissolved in our saliva. It's going to enter through the taste pore, and it's going to be detected by our gustatory hairs. And then the gustatory epithelial cells are going to translate this chemical input into an electrical one that's going to be processed by the brain. Now, these gustatory epithelial cells can detect 5 basic taste sensations, depending on the chemicals that are going to be dissolved in the saliva. We have sweet from carbohydrates, sour from acids, especially hydrogen ions, salty from metal ions and salt, as in sodium chloride, and bitter from alkaline chemicals like caffeine. And we finally have umami, which is found in amino acids from meats. As you can see, spicy is not a taste because spicy is detected by pain receptors, which is why when you eat something that's spicy, you feel a burning sensation. It's because it's felt as pain. People that can eat very spicy foods, they have numbed these receptors and they don't feel the burn like regular people do. Now, some interesting things about taste is that there are taste buds in the back of our throat, so you can literally taste stuff on the back of your throat. And also that it is 80% stimulated by smell. So it is true that when you have a cold and you can't breathe properly, food is not going to be as tasteful, and it's because it's influenced 80% by your smell sense. The sense of smell is fairly similar compared to the other ones, and this is because our receptors are actually going to be exposed to nerve endings called olfactory cilia. The way this works is when you breathe in airborne chemicals, they are going to get stuck on a mucus layer that's covering the cilia, and then they're going to bind to the olfactory cilia, which are going to detect these chemicals, and they're going to transform the input into an electrical one that is going to reach the brain through the olfactory tract. And this all happens through a secondary messenger system. Also, I don't really like this image, so I would recommend you to look at page 576 in your book. It's got a way better representation of this olfactory cilia and the structures involved with the smell sense. So this video is getting extremely long. I thought it was gonna take less time. My goal was to try to touch on the basics of how each of the senses work, but it's already at around 10 minutes. And I don't want you guys to have to sit through a 20-minute video. So I will cover hearing and balance in a different video that I will post later this week. So you can focus on these 3 senses first, and then we can focus on hearing and balance, which are a little bit more complicated. Also, if you have any questions from this video, you can come to Wednesday's SI session and ask me on April 8th. I will probably be posting the hearing and balance video on either Thursday and Friday after that, and we can discuss hearing and balance on Saturday, April 11th. And I will still be posting the review video from all the chapters on April 12th. I will try to keep that to the point so that it doesn't become extremely long. So it's going to be very vague and just touching on the most important points of each chapter. And we can have the April 13th session to review all the chapters.